Hi, welcome everyone. I am Dr. Alan Bonder. I'm one of the associate editors for CLD, and I had the pleasure to have Alanna Strong with me today, which she is going to discuss her most recent paper that she published with us. First of all, I'd like to introduce Alanna Strong. She is a pediatric geneticist, and it's a pleasure to have you here, Alanna.
Thank you. It's a pleasure to be here.
All right, Alanna. So kind of the first thing that I want to know is just remind me, or give us a kind of a brief summary about what your paper is and what's the impact of your paper in liver disease.
Yeah, so the paper that we're discussing now is the urea cycle paper, which tells the natural history of urea cycle disorders and how they're recognized initially and how they're treated and managed, and the importance of being able to correctly identify a child or an adult that has a manifesting urea cycle disorder to prevent any future morbidity and mortality, and also a little glimpse of potential future techniques that we have for earlier detection to prevent further morbidity and mortality, and also for treatment.
So let me ask you some specific questions about your paper. So why do virtually all living cells produce ammonia?
So it turns out that ammonia is actually a natural byproduct of protein metabolism. So when we need to do the things we do to survive, all of our activities of daily living require energy. And humans and most other organisms get their energy in the forms of fats and carbohydrates and protein. And when you break down protein, you have to break down nitrogen and it forms ammonia. And we need to have a way to get rid of this ammonia. And so that's why we have a urea cycle.
All right. So what's the problem? So why is ammonia a problem in the body? So what happens if we don't get rid of it?
Yeah. So ammonia is unfortunately a very potent neurotoxin. So it can cross the blood-brain barrier, go into the brain. It can cause swelling. It can cause altered mental status by interfering with neurotransmitters and neurotransmission. And so it's extremely important that we keep the levels as low as possible, below 50, as the typical normal level in adults, just avoid that neurotoxic effect of ammonia.
So when we talk about kind of the urea cycle defects, can you just tell me what does that mean? So I mean, I'm just an adult physician. So tell me how does that translate in someone who has a problem like this?
Yeah, great question. So different organisms have devised different ways to get rid of ammonia. And the decision of how to do this is based on what you have more of. It's a trade-off between the amount of water that you need to conserve and the amount of energy that you need to conserve. And so for organisms like humans that have an intermediate amount of water and an intermediate amount of energy in order to be able to get rid of ammonia, we get rid of our ammonia by converting it into urea, which is a process that takes some energy and some water, but not at the extremes of all of them. And so there are multiple genes that encode enzymes that help in the process of converting ammonia into urea. And there are also a number of transporters that help move different intermediates in the step at different points in the cell where these enzymes can act. And a urea cycle defect is a loss of any one of these genes or transporters that interferes with the integrity and proper function of the urea cycle.
So what is the most common genetic mutation, or what are the most common conditions that will affect the urea cycle? Or what's the most common diseases?
Yeah, great question. Yeah, so the most common urea cycle disorder is ornithine transcarbamylase deficiency, or OTC. That's an X-linked, I'm using the word recessive because usually it's more severe in males, but females can manifest, often do manifest, and can have very severe disease. And that's one of the conditions that I really want to highlight, because often missed in females until they either have a catastrophic event as an adult or have the stress of childbirth, which is an extreme protein load, given the extra blood and the extra growth of the uterus and the rest of the tissue. And it can decompensate if it's not recognized. And so that's a really common urea cycle, both in children, also really important for adult providers to recognize in the female or even the male who, under very stressful conditions, develops high ammonia or altered mental status.
And how do we treat the defects? So what do we do to make them better?
Yeah, great question. So unfortunately, right now, there's nothing that we can do to make it better. We can't cure it at this time, with a rare exception. But what we do now is we control control the amount of load. So as you know, the driver of ammonia that produces the need to make urea is protein. And so these children, once they're identified, are put on a low protein diet to minimize the load on the urea cycle, and they get their calories from other sources. We also do our best to provide medications that provide alternate routes to get rid of ammonia without using the urea cycle. So these are nitrogen scavengers that can complex the nitrogen-containing molecules and also ammonia to help individuals affected by these disorders pee out their excess nitrogenous waste without relying on the urea cycle.
I think you already mentioned what will be the treatment. So what is the role of liver transplantation in these defects? Are patients cured, or what is the outcomes of patients who have a urea cycle defect and undergo a liver transplant?
Great question. So right now, the urea cycle is most active in the liver, also done in the kidney, but the liver is the primary site. And so by replacing an individual's liver, you are functionally replacing their impaired urea cycle. And so it's essentially curative. So individuals who undergo liver transplantation can have liberalization of their diet. They don't usually need nitrogen scavengers anymore, and they can go back to not having a urea cycle disorder. The limitation of transplant is that, unfortunately, if an individual affected by a urea cycle disorder has had a catastrophic hyperammonemic event and had consequent brain damage, that is not reversed from the liver transplant. And so you can cure the urea cycle, but not undo the neurocognitive effects of anemia.
So is there any kind of future treatments? For example, are we looking, for example, genetic transferring treatment options? Is that an option, or is that something that is that something close to, the future, or can you comment a little bit about that?
Yeah, absolutely. So right now, in terms of future directions, there are two things that we're focusing on. If we can't cure it, how do we prevent the morbidity and mortality from the disease? And then, of course, how do you cure it? And one of the big things that we're focusing on now is trying to capture individuals affected by urea cycle disorders in the newborn period before they have a catastrophic hyperammonemic event, so we can prevent the brain damage. Right now, most urea cycle disorders, most notably ornithine transcarbamylase, is not detected on the newborn screen. And so what we're trying to do is expand newborn screening either through sequencing or through different biomarkers to be able to diagnose these children earlier and start treatment earlier. In terms of gene transfer gene therapy, definitely has been very successful in animal models using viral vectors, both AAV and adenovirus. The human trials, unfortunately, have not been curative yet. So right now, we don't have an active gene therapy that's successful for urea cycle disorders in humans, although this is an active area of investigation.
And I think you bring up a point about what to do even before the patients get sick. So can you comment a little bit kind of the screening test, or how do we screen a newborn for this condition? That there is a standard of care to screening the newborn for every condition. Can you comment on this?
Yes, absolutely. So right now, all newborns born across all states have a newborn screen, which is a heel prick after 24 hours of life that tests for a whole host of largely genetic conditions, some non-genetic, that if you identify these children immediately and early and initiate treatment, it prevents morbidity and mortality. And right now, the biomarkers that are being used that are relevant to urea cycle disorders are plasma amino acids. Measure the amount of each amino acid in the blood. It helps detect things like maple syrup urine disease, which is not a urea cycle disorder, but something that people think of with amino acids. But in certain urea cycle disorders like citrullinemia and arginine deficiency, you can have differences in the arginine concentration and in the citrulline concentration that can be detected on newborn screen. The newborn screen is flagged. Children are brought into the hospital, confirmatory testing is sent, and ammonia is measured. And if the ammonia is elevated or if confirmatory testing is positive, treatment is initiated immediately, so in the pre-symptomatic phase. Unfortunately, with OTC, you don't always see plasma amino acid disturbances. And so while some of these children can be caught on newborn screen, they often are not. And so the hope is that through the introduction of sequencing on the newborn screen, which actually proofreads the genes, we can avoid missing children by not focusing on the biomarkers, but instead focusing on the actual gene.
So let me get back to treatment. So kind of early detection. So is the early detection going to prevent the need of a liver transplant and the need of further treatments?
Perfect question. So I think the decision to undergo liver transplant is very individualized. So some families are very scared of the possibility of a hyperammonemic event causing irreversible brain damage. And sometimes you can do everything correctly there's still a hyperammonemic crisis. A child gets an influenza infection, a child gets COVID, and they were controlled well their entire life. And now all of a sudden they have a metabolic decompensation. The ammonia can rise extremely quickly, and you now have irreversible brain damage as a result, or the potential for. And so for many families, the decision to undergo liver transplant is I want to do this immediately. And in terms of for families who don't want a liver transplant, if you're able to catch these children early and initiate special diet or a low protein formula. And also one of the big things is when children get ill or when they're not eating well, it also creates a protein load through muscle catabolism. And so if you can avoid that in the newborn period by providing dextrose-containing fluids in the context of illness and bringing the children into the hospital, and also in the newborn period, if a child is breastfeeding and isn't able, the mother's milk supply hasn't come in yet, identifying those children and just supplementing them immediately would be really helpful in preventing a hyperammonemic crisis.
Excellent. Well, Alanna, thanks for this very detailed explanation. It was a pleasure to have you here, and really I'd like to thank again for your contributions to our journal. So I don't know if you have anything else to add at the end.
No, thank you so much. And I think just the most important thing to take away is that although urea cycles are thought of as a primarily pediatric disease, not to be seen in the adult clinic, I would encourage you that if there's ever an adult who has altered mental status, to check an ammonia and really keep a urea cycle disorder in the back of your mind, because while the treatment for hepatic hyperammonemia it can be effective in individuals with a urea cycle disorder, treating the underlying urea cycle disorder can actually decrease morbidity and mortality and decrease the duration of hyperammonemia, which is very good in improving outcomes.